Hi, everyone. Thank you for joining. As usual, we're going to
Hi
take a
everyone.
couple of minutes. Yeah, we're going to take a couple of minutes and let everybody join. But maybe in a few minutes, we will start with like a warm-up question, just in case. But yeah, we can wait a few minutes till we have more people. I'm going to also send a message on the Slack that the waiting room is open. Okay, maybe actually I'll go ahead and I could type it in the chat, but I'm curious if anybody has any questions about the homework or what we went over last week. Or about from the first week, like basically kind of any question, yeah, maybe related to course material. Feel free to unmute or you can type it in the chat. Okay, maybe no questions from last week. That's also fine. Yeah, you can of course always go ask a question on Slack. I hope you enjoyed the homework. I thought there were some fun questions in there. Hopefully they actually were fun. Yeah. Oh, I see. Okay, yeah, there's some stuff in the chat. Okay, awesome. Okay, so someone said they they didn't like, okay, the last question that asked you to look up the iGEM parts. I'm wondering actually, I think this is Nelson, if you could elaborate, was the problem that you didn't want to go to the website, or was it that it was unclear what you had to do? And did it become clear later?
Can you hear me?
I can, yes.
Actually, I think it's maybe the way the question was a bit framed.
Mm.
But yeah, I could possibly do it, but maybe I didn't get the explanation more. But I got the point. Got the point.
Okay.
But now I cannot access the question, so I don't know. Maybe if you could have it there, you can use it as an example.
Ah.
Yeah.
Yeah.
I don't know if the deadline has already passed. Yeah.
Yeah. No, so actually a couple of things. So you can you could like the okay. So the homework is due now, basically. So the people who grade the
Sure.
homework finished it or sorry, it's not due now. It was due last week. So I think when it was turned in, it should have been graded. We know like a lot of people have joined the course late. So basically what's going to happen is the grades are going to be released today, and then you'll get to see the answer key and the explanations for sort of what type of thing we were looking for, which may be helpful for I guess you didn't understand the question. Also, though, like we're trying to make writing our questions better. So if something is unclear, we like it if you tell us. But yeah, I think everyone should. I don't think we've released the grades yet. I think I'm going to press the button at the end of this. But please go back and look at you can look at the answer key and it should have a fuller explanation. But yeah, the hard questions are a little more open-ended, so apologies if
it was a bit okay, cool.
Awesome. But yeah, thank you for yeah, engaging.
Okay. Thank you.
Yeah. Yeah, that's a good point. I guess like you haven't received any graded homework back yet. So today will be the first time that if you submitted a homework, you'll receive it graded. And a couple of things that maybe you saw, I don't know if we stated them explicitly, but you can actually get more points on the homework than are necessary to pass. So if you don't want to do the hard questions, you don't have to, but we think it's kind of a useful challenge if you want to push yourself. And if you try and you only get partial credit, that's fine, you still will get partial credit. Also, we understand that like people are taking this class sort of as a hobby. So or like as a part-time thing. So you might not be able to attend the course every week. You might not be able to turn the homework in on time. So you can actually turn in the homework whenever you want. Just the thing that will happen is it might take us a while to grade it, which means it'll take a while until we issue your completion certificate, but that's fine. You can get the same thing just later. So please do what works for you. I will say though that it'll get harder to keep up if you don't stick with the deadline. So I really encourage you to like get the most out of the class, like to kind of follow it at the pace that we've set. But if your life needs some more flexibility, that's okay.
Okay.
Yeah. Are there any like maybe like
there's another
logistical
question on the chat box.
From Sarah. Yeah, thank you, Nelson. I actually was just going to add quickly, if you have any other logistical questions, maybe also feel free to put those in the chat. So Sarah has a question, please elaborate more on protein domains. So I guess I'll start to answer it a little bit in a general way, but if you need a fuller answer, Sarah, you can type more in the chat. But basically, when we talk about protein domains, I think it's a way of talking about subsections of the protein sequence. And usually these subsections are either like they fold, I guess, together and are kind of a different part, or they're defined because they have a distinct function from the rest of the protein. So one of the ones I like to think about is like a receptor that's like on the cell membrane. It might have what's called an extracellular domain, which is the part that's outside, an intracellular domain, which is inside, and then the transmembrane domain, which is kind of crossing the membrane, right? Because it takes some distance to cross the membrane. And what's interesting is those domains will have maybe different chemistry and different function. For example, the transmembrane domain is going to be around there's this, like the plasma membrane has both hydrophobic and hydrophilic parts. So maybe the transmembrane domain needs to have chemistry compatible with that. The extracellular domain might want to bind to something in the environment like a sugar, whereas the intracellular domain is doing something like activating a downstream response. Domain usually refers either to a fold or a function. And as we talked about, those are sometimes basically the same thing, but it's a way to break up the protein into parts. And it's also a way, I think, of making proteins more like Legos. So for certain types of protein engineering, you can just assemble these domains in particular ways and then get a new function. And that's sort of what question 5 on the homework was trying to get at, is this idea of like, just imagine like the different type of Lego creations you can build. Although like often it can be as simple as putting 2 things together. I feel like often in our group meetings, people have some design and they'll be like, oh, I realized I needed this, so I put a degradation tag on it, or I needed it on the surface, so I put a transmembrane domain on it. And so I think it's a common thing that we like hadn't talked about in the beginner course, and I thought would be useful for you guys when you're thinking about things you want to engineer. Hopefully that's helpful. Okay, so I wanted to add, so Kato is usually here to welcome you guys, but he says his network is unstable, so he may or may not join. But I'm gonna go ahead. I feel like we've reached maybe enough people joining. Oh, Nelson has a comment. Synthetic biology can be tackled with bioinformatics tools as well. Yeah, certainly. There are actually bioinformatics tools that are useful for, predicting folds and also identifying protein domains. Yeah, awesome. Yeah, thanks, Sayandeep. Okay. Actually, we still have some people coming in. Okay, I have a quick presentation where I want to talk about something not quite related to homework, but the final project, which is something we added to the intermediate course because we'd like you to do something a little more creative and a little more challenging. So I'm going to share my screen and let's see. So, okay. First, I wanted to just say quickly that we've been adding people to the course because some people signed up late. I think I even exchanged emails with a couple of people who are joining for the first time today. So welcome to the class. We're like super happy to have you here. We're excited that we can, have as many people as possible in the class. But also I just want to encourage you, like I said, something happens that's kind of significant every week. So you may have missed something. So if you go to the syllabus, which we can send a link in the chat, it has links to the old lectures. Basically in the syllabus, there's a part where every week is a section header. So you can click on the week and then you'll see a link to the slides, the lectures, and then also the homework. And something that's important to sort of complete the class, if you're interested in getting the certificate, you don't have to be, that you kind of complete all the homeworks. So if you're joining today for the first time, we definitely encourage you watch the lectures to sort of get all the material. And if you want to go back and do all the homeworks, please do so as well. Okay. And all right. So, but now I want to talk about the final presentation, which is like, what are we doing and why are we doing it? Okay. So the final presentation is basically going to be a combination of some important social problem and a synthetic biology solution. Because I think a lot of what motivates why we're here, and I think why Sayandeep and I do what we do, is because synthetic biology is a powerful tool to understand the world and also create new technologies. It's something we would like you to complete as a group. I know that many of you are located in the same city, but some of you are by yourself. But because we're doing this online and we might be able to help out giving you Zoom links and things, we'd like you to collaborate together. And basically, it's going to have 3 pieces. The big piece is going to be a slideshow presentation where you talk about the problem that you pick and what the solution you have come up with, the synthetic biology solution that you've designed. Something else that's also going to be a key part is answering audience questions after the presentation. I really want to encourage people to ask either just to clarify to understand better or to ask something about like applying the design differently. So you can practice both presenting but also answering new questions. And then the last thing is giving feedback on each other's presentation. So each of you will have like a form that you can basically fill out to grade another student. And part of the idea is here, I think by watching other presentations, you get basically really cool ideas for like how to do things better or like something that you liked. And so we think that'll be important. Okay, so that's kind of an overview of what it is. And so why are we doing this? So I think one is to synthesize and solidify what you guys learn. So a bit like we're talking about putting like protein domains together, like the topics that I teach about and then Sayandeep taught about and then I taught about can actually all be put together to create a full project. Like Sayandeep's going to talk about strain engineering. Like without that information, you don't know how to realize some of these protein engineering designs that we talked about. So we want to give you a chance to see what it would look like to sort of put these things together. And also because mostly we assess what you know by like grading your homeworks, but we don't know exactly how you do it or how easy or how hard it is for you. So I think we'd like to see at least a couple of you presenting and talking about what you know as a way to really see how deep your understanding is. And also, oh, sorry, Kato has his hand raised. Yeah, Kato, do you want to add something?
Okay, thank you so much, Heidi. I hope you can hear me now.
I can.
Yeah, what I'm suggesting is that the students also should take this presentation and the final project as a major issue because they can help them, like if they decide to participate in international competitions such as GOJEC and maybe other competitions which SynBio4All Africa will organize in the future. This will be really baseline to help them have the experience to participate and maybe compete in those competitions. Yeah, thank you so much.
Yeah, thank you for adding that, Kato. I wanted to bring that up later. So Kato, I think, founded this organization as part of his participation in one of these competitions. And like, in addition to this being something cool you can do for the course, it can definitely, you can take this project and keep working on it and present it at something like iGEM or GOJEC. Or what we'd really like to do is have a competition run by SynBio4All. Yeah, I think it can be useful for you. And I guess like kind of related to that, I think the other thing that we thought would be fun is like just to practice other types of skills that are important, like being creative in terms of coming with a new idea, thinking about how to define a problem that's often as hard as coming up with a solution, and working together in a group. Okay, and if you're thinking, oh no, this sounds really hard, here are a list of a couple of things that we are trying to do to make it more feasible for you, because we realize we're trying to build you up to doing this, more challenging thing. And so first of all, we've designed a rubric, or basically a recipe for how we're going to grade you, or like, and what the essential components are. So if you sort of add all the elements of this recipe reasonably well, you will be fine. Also, like the homework for the project, we have more points available than you need to pass the class. And in particular, that like part about giving feedback on other people's presentations, if you do that well, you already have a third of the points you need for the project. So if you struggle with the project, if you can give other people good feedback, you'll be in good shape. You're gonna have 2 weeks to put together your final presentation. So the course is 10 weeks. But the last 3 are all related to the project. So there's no lecture on weeks 8 and 9. That's like time set aside for you to meet with your group and put together your presentation, although probably it'll take longer than the 3 hours that would have been the class time. And we encourage you to come up with your own topic. We think that could be really valuable, but we're also going to have a list of topics for you to choose from. So we don't have them yet, but they'll be available on the syllabus next week. So you can just pick something from there. Okay, so here's just a quick overview of our recipe. As I said, there are these like 3 parts. So in the presentation, there's basically what we'd like to see is that you describe the problem that you want to solve. Basically, like, why is your project valuable? Your solution should be accurate in the sense that it's like actually something that would be using synthetic biology and not something else. And appropriate in the sense that you can explain why this solution is actually, why this will actually be helpful for this problem. Talk a bit about advantages and disadvantages of your design, maybe like what are the pros and cons relative to other approaches. Also somewhere, this could be related to advantages and disadvantages. There should be some comparison to a competing technology. I think there are this is like something that's really important right now, and I think hard in the field, is thinking about, okay, I could engineer a cell to do that, but should I? Is there a better way to do it? Or even like, should I use a cell-free system instead of engineering a whole cell? There are reasons that like different approaches would be better. So that's kind of the analysis we'd like you to do. There should be a section on biosafety in terms of thinking about, is your solution like safe, or what are the risks involved? And then also responsible conduct of research. So we'd like you to correctly cite your sources and don't abuse ChatGPT. And we haven't talked about ChatGPT in the course. I already saw on homework 2, somebody had copied and pasted from ChatGPT their answer. So I know it is happening, and that's not really the kind of learning we want to do here. I know that ChatGPT can be useful for like explaining things and accessing stuff, or maybe even like writing some good English. So my fiance is actually not a native English speaker, so he uses ChatGPT to improve his English when he's writing. And I know many of you are also non-native English speakers. So I think there are ways to use these tools that are valuable, but please don't just like have it generate everything for you and copy and paste. Although you will be giving a presentation, so it won't quite be the same. Okay. So in addition to the presentation, then there will be a question and answer section. So that's just like people are going to ask questions afterward and you answer them. And then also peer feedback. So we're basically going to have a form, which is like a set of questions that you fill out and give people feedback on their projects. And this will be great because this means that you also get feedback again if you want to take your project somewhere else. Okay, this is our recipe. Okay, and then some quick advice. And these are also kind of in the syllabus, but I encourage you to start brainstorming project ideas earlier. This is something you can do individually, just like keep an eye out. Maybe there'll be things that you encounter and you're like, oh, that would actually be a good project. I think I would like you to, if you can, pick something that excites you or that matters to you. I think it can be more fun to work on something that you picked yourself. Try to pick a problem that can be solved by synthetic biology, or like synthetic biology actually makes sense. Expect that problem solving is going to be nonlinear. Like just because you've picked the problem or you think you have a solution, you may get kind of into it a little bit and then find out that, oh, this isn't quite right, or maybe this is a solution to a different problem. But feel free to give yourself some time. Like I say, you use like basically 2 weeks to work on the project. Maybe in the first week, you're kind of figuring out if your solution makes sense. And then in the second week, you're refining it and making it better. And we'll be available to answer questions if you feel like you're stuck. The other thing I wanted to highlight or sort of say is like, as much as the project feels ambitious, I think if you can make it small for yourself in a particular way, that can also be good. So having a well-defined problem is better than picking a big problem. So for example, one of the ones we have in the project bank is the idea of I think it's about a lake in Ghana that has like this invasive plant growing on top of it. So that's basically a lot of biomass you could use to convert into something else. But that's like a very specific, well-defined problem. That's a bit easier to work on than saying like, I want to recycle all the plastic in the world. Like that's a much bigger, broader, and harder problem. Also, well-explained solutions are better than very creative and crazy ideas. It's okay if your solution feels a little bit maybe simple to you, or maybe isn't as like fancy as you would like. What we really would like to see is that you understand what you are proposing to do. And similarly, something I've seen when I've judged other types of like science fairs or science Olympiads is often what we're looking for is people can explain what they did, not that it's a perfect solution. So we don't expect that your report is like definitely going to be a good solution. So if you come up with an idea and you start explaining it, and then like you're like, I think in the presentation we should talk about like these key challenges. That's great. I think if you can show that you can be critical of your idea and you've thought about it carefully, that's more important than the idea being perfect. I also encourage you to look at example projects. So we're sort of modeling, go back. Oh wow, too many meeting controls. We're modeling some of basically this project over a particular category from this GoGek competition. So this is the link. It will take you here where they show the projects from, I guess, earlier this year. So some of the projects are experimental. They did experiments. Some of them are computational. They did things on their computers. And then there's a proposal category where people proposed an idea. And so this is what we're going to do, because you don't have to do any experiments. You don't have to run any simulations on your computer. We just like you to explain the problem. And of course, ours is a little bit different from theirs. They made these posters. They had a written report. So that's a little bit different. But this could give you a sense of how people picked a very specific problem and then defined a very specific solution. And also just kind of get inspiration for the kinds of problems people are working on. Okay. All right, so sorry, I'm being a little long-winded, but this is all the advice. Okay, and then just quickly timeline. So the reason we're talking about this today is because we sort of start working on the project now, in the sense that it's time to form your groups. So we would like you to talk to each other, like you can message each other in the chat on the Zoom call, but also Slack could be a great place. If you just wanted to message there and say, hey, I'm located in this city, or hey, I like to work at this time, or hey, I want to do a project on like this particular topic, who's interested? You can do whatever you want to sort of assemble a group of people. We do say that the max group size is 5 students. And I didn't mention this before, but larger groups will give longer presentations. So it's okay if you have a small group. We don't expect you to do as much work as a large group. Whereas if you're a large group, we do expect you to like think about it a little more deeply, but we could talk about that. But so you're going to submit your group's email addresses as part of homework 3. And we're using the email addresses just because it's a little easier way to keep track of people rather than your names, which maybe you type slightly differently, or maybe you like don't include your last name, or you do include your last name. So please use your email address. Use the same email address you used to submit the homework, and that'll just make it easier for us to write scripts to sort of just check what the groups are and put everybody together. So please put your email addresses. And remember, you're submitting this as part of homework 3. That means that's an assignment that's available today and due in a week. So basically you have a week to find your group. That also means that by week 5, when the homework is graded, you'll know if you're in a group. If you're not in a group, you can basically submit a request to form a group, and we'll help you form a group in week 6. But that will come up then. And there is also an opportunity to do it individually, but we really encourage you to find a group. Week 6 is also when you should start brainstorming your project ideas, because homework 6 is when you're going to put in your project ideas. And here we just want to look at the project ideas and give you some feedback to be sure that, it's specific enough. It seems like something that can be solved by synthetic biology, and maybe also to just give you a few hints about where you can start. So that's going to be submitted as part of homework 6. That means that by the time you get your homework back, which is week 8, you'll know what your topic is, you'll know what your group is, and then you can do all these things together. And then you have 8 and 9 during class to work together. And then during week 10 are the presentations. Okay, sorry, I see a lot of messages. Okay, so everyone's messaging about looking for a group. Okay, great. Wow, some people already have groups, which I am like very shocked. I would not have a group yet. I would be someone who's like, oh no, I'm gonna message on Slack now. So yeah, please feel free. And then we're happy to help if you need help. Okay, I think, okay, so that's actually the end of it. Wow, it's already 1. And I don't know how much material Sayandeep has, so I don't want to take away from him. But let's just say, so if you have any questions, maybe please message on the Slack or send an email. We can also discuss more next week. The main thing is I just wanted to give you a vision of what's coming. There's all of the details around the syllabus. So this is a link that you can just, sorry, if you, we'll share the slides, but if you click on the link, you'll go here, you'll see there's a whole section called Final Project, where I basically say the same things I said before, but in more detail. So I encourage you to go there. But the main thing is just that I wanted you to know you're putting your group together so you can kind of do this thing together. Okay, that's everything I have. Yeah, thank you guys for listening. Good luck finding your groups. And yeah, maybe you can hit a pause on it so you can actually listen to all the really helpful things that Sayandeep is gonna teach you. But yeah, good luck. And yeah, I think you're in for a treat today.
Thanks, Heidi. Thanks for the wonderful intro to the class. And hello, everyone. I'm hoping that you guys have got some time to go through what Heidi and Hari taught you guys about the nuances of synthetic biology, about the nuances of protein engineering. How do we do that? What kind of philosophy, or I should say, what kind of principle we actually rely on while doing engineering. So today we are going to talk about strain engineering, engineering microbial strains, be it a bacteria, be it yeast, be it something else, but any form of microbial engineering that we can do, and why we need to do it. Why cannot we just engineer a protein and put it back inside the cell that we want to? Why we need to engineer a strain? So that's all we are going to discuss today. We will be discussing some principles of doing the work in real life, and there would be some examples of how we do it, and there would be a short discussion on some of the tools that we use in the lab. So do not put too much of the stress on learning the tools in like the most extensive ways, because the tools are ever changing. And till now, synthetic biology tools, there are some tools, of course, which are like age-old tools. We rely on them, but most of the tools are evolving. So it is more important to understand the logic, the principle of their working mechanism, so that you can apply the knowledge, you can apply the mechanistic knowledge in realizing any tool, utilizing any technique that it might fall on your lap. So without much further ado, let's start with that. I need to share my screen. Okay, and I'll do that. Who? Can everyone see my slides?
Yeah, it looks good.
Okay.
Oh, now I see your presenter notes.
Okay. I think, I don't know. It's fine. Sometimes it's confusing. Let me stop share. Okay, I think I'll be happy with all of these things. Fine enough. Let's start with that. So, strain engineering and validation tools and techniques, and this is the topic of our lecture today. And before we start this, we need to understand, why do we need engineered strains? I mean, just as I was talking before that, I mean, you could have done with something else, right? I mean, use multiple organisms. Why do you need to engineer them? I mean, a bacteria, say for example, it can do multiple magics. Why do you even want it to do something for yourself? So before we start with that, I would like to begin with a weird story. It might sound very weird, but let's talk about cannabis. Why cannabis, if you are interested? Well, because naturally occurring cannabinoids are used for treating a multiple array of disease symptoms. It can be epilepsy, it can be chemotherapy, associated nausea and vomiting. And so most of the cases where you have a drug or a medicine which employ a lot of pain suppression, which imply a lot of neural suppression, they use a lot of opioids or cannabinoids. So what we have in real life is some researchers in UC Berkeley back in 2019, they engineered the yeast cells to produce 4 major cannabinoids from simple sugar galactose. And this sugar is like always present in the yeast medium. So you can just supply a very simple form of this galactose and get the cannabinoids. Is it natural? Of course not. So what is natural in yeast is the mevalonic acid pathway. And the researchers did exactly what the researchers should do. They took the mevalonic acid pathway. They recognized that some of the intermediates can be pushed towards cannabinoids production. And they did that by genetic engineering and metabolic engineering. And voila, we have a whole heterologous biosynthetic pathways that are introduced by new genes from other organisms. So it's not just about the yeast genes. They took genes from other organisms, they put it back into the yeast. So if we can produce anything, hi, good luck. Are you saying something? I guess not. Okay. If we can produce anything, like if we can produce cannabinoid, we can produce anything, right? So what about strains that can eat pollutants, induce crop growth, deliver drugs to tissues, produce nutrients, target disease cells, and store data in DNA? So we can do all these sorts of magical stuffs with the strain engineering part. All we need to understand how to go on with that. So the first thing that we need to learn is about the engineering cycle. Probably many of you are already aware of it, that the engineering cycle is commonly known as the DBTL cycle: design, build, test, and learn. So design is where you start your work. You design the thing that you really want to do. Build is where you actually use the tools, use the techniques, and go in the lab bench, start making something. Test is where you want to know whether your design is 100% correct, whether whatever you wanted to achieve, are you achieving that? And learn is all that you can take away from test. So basically, it's a never-ending cycle. What you learn from the learn cycle, you put it back into the further design cycle. Your design, most of the time, would not be 100% correct at the very first attempt. So you need to know what to take as a lesson from the learn cycle, and then go back to the design cycle, doing more and more rounds of things. So it's a very iterative process. So as you can see in the figure, it's a very self-explanatory figure. Let's say you have started with a cell, which is your initial design. You start with that, and your first designs are based upon the initial organism, right? If you are choosing E. coli, you have a whole lot of genetic setup for E. coli. If you are choosing yeast, prokaryote to eukaryotic transition, your genetic setup changes. So your start of the design is always based on your initial organism. And then you build upon that. You you make a design. Okay, I want to change this particular promoter so that I can have protein X produced in such a way. You change that, you do everything, you calculate, you sort of draw a figure, sit with your computer, make a whole computational program that will help you to understand what are the translational efficiency you should achieve, what is the transcription efficiency you should go for. Everything that is part of your design. And once you're done with the design, you make the clones. You go with the cell, you put the clones inside the cell, you build something. Now you want to test that, whether your proteins are getting produced in exactly the way you want. How do you test it? You test it with different kind of parameters. You want to see whether the protein is getting expressed in cytosol or outside the cell. You have tools for that. And so you do that. You take the outside environment of the cell, look for the protein. You take the inside of the cell, you look for the protein, all sorts of that stuff. But what if you see that your protein, let's say you started with a design that says protein X would be secreted outside of the cell. But when you are at your test cycle, you could see that not all of the protein X is getting out of the cell very easily. Some of them are getting retained by the cell. So you're not getting 100% productivity as per your design. You then go back to the design cycle. You try to identify what went wrong and you redesign the whole thing. So that's all about the engineering cycle. So if there is an engineering cycle, are there steps in the cycle? Of course, there are. So the steps are, as you may say, the different strategies or different phases of an engineering cycle or the strain engineering itself is such as this. So first is the chassis selection. You need to select an organism, your chassis, your starting material. It can be anything, but you need to select it based on what you are trying to achieve. Then comes the process of pathway designing and selection, and then the actual strain engineering with the help of genome engineering, metabolic engineering, and all those stuffs. And then comes the part of screening and validation. So you can say that the chassis selection and pathway designing is all about the design cycle. The strain engineering itself is the build cycle, and the screening and validation is the test and learn cycle. So that's great. So let's start with the chassis. So what makes a good chassis? And the question is very important because you don't want to be starting a project picking up a wrong chassis. You want something to work inside the human, and you are not worried about its production, high production or something. You just want to have it as in a better functional way. You're not going to choose an E. coli, are you? Because if you want the protein to be expressed in E. coli, you have to redesign it in bacterial expression way. Maybe you want to choose an yeast cell. Maybe you want to choose a mammalian cell as an intermediate, but maybe not a microbial cell. On the contrary, if you are really, really concerned about the protein's production, you want it to be produced in a very high amount, and you know that you can go to the human cell after some time, not right now. Bacterial cell is your first choice. So basically what you did here is that you looked at the project, you basically stepped back and looked what are the steps behind the whole project, and then you chose your chassis. So this is what we do in terms of chassis choosing. And also to identify a chassis, it is very important that you are identifying an organism about whom you know a lot. Because if you don't know a lot about the organism, if it is a novel organism, you need to standardize it first. You need to know its genetic tools first. What kind of a genetic setup it does have. Does it show a very slow growth rate or a very high growth rate. You need to know all of these things. So that is not doable. I mean, that's gonna push up the cost and also the time, right? So you don't want to do that. You start with the model organism about something you know already. So these things, whether you know about their genome, whether you know about their physiological characterizations, whether you know from the literature that it is easy to knock things out of their genome, all of these things are really, really important. And also one more thing is very important is their metabolic models. There is something called a flux. Maybe you would be hearing about this in the next class, which is on the metabolic engineering. A flux is the gradient of the metabolites at different stages of metabolism within a cell. So that is in the crudest sense of the term. So why do you need to take care of flux? Because sometimes it happens that if you want to change the protein Y inside the cell, but you never know whether changing protein Y would affect like a change in the production of protein X. And if it so happens, maybe a change in the production of protein X being produced in a low amount, slightly low amount, is very lethal for the cell. So you need to know all of these things very carefully. So choosing a chassis, that's very important. And here you can see there are some intended applications, like if we are to go for harsh biochemistry, go for protein production, going for some light-driven production, what are the natural features and traits that we should look for then? If we are going for harsh biochemistry, we should go for a chassis which has a lot of chemical resistance. Pseudomonas putida, for example, it has a very good application in bioremediation, in eating pollutants. So that's a very good example of chemical resistance. Bacillus subtilis, for example, it has a very well characterized secretion system. So if you want your proteins to be secreted outside of the cell and you want it to be in a very high amount, Bacillus is your choice. A variety of tools and knowledge about the genome and the proteome. E. coli, without any doubt, but none of them can do photosynthesis. So if you want to do that, you want to go for cyanobacteria. So all of these are there. But if you look closely, can you see a common trait in all of them? Reduced genomic strain. And that is very, very important. Why that is important is that a reduced genomic strain gives you the advantage of focusing only on your engineering part. If they had a huge amount of engineering strain, you wouldn't have been able to, you would have been busy in rectifying one thing after another in their genome, and it would have been very difficult, time-consuming, and not at all cost-effective for you to engineer those cells. So the features of an engineered strain is to have a reduced genomic strain, have a predicted flux distribution, something that you can vouch for, easy control of gene expression. You want to make all the modulations in the genome, right? So you want the easy control of gene expression, one inducer molecule, and everything is under control. You want to have the total manipulation on the protein production. And last but not the least, systemic scaling up. Why? We will discuss this in detail once we are in the test cycle and launch cycle. But as of now, keep it in mind that whatever you do on your lab bench has to be there out in the industry. So you can do something really wonderful in the lab bench, but until and unless it can be scaled up in a way so that it can reach people, it can reach the mass, what's the use of that? So systemic scaling up is very important for strain engineering. Of course, if you are trying to do something, if you're trying to engineer a tool or a technique inside the cell, you don't have to think about the systemic scaling up, because then the cells would take care of that. And some people at some other research level will think about the systemic scaling up, but that is an important part of the thing. And the general approaches for creating the synthetic microbial chassis is usually two, top down and bottom up. So before we go to top down and bottom up, here is a quick challenge. You don't have to do a lot of maths or something, just take a quick guess. How much do you think can be cut out of the E. coli genome and still produce a viable strain? In the chat? 53%. Okay, 53. Oh, wow. 26. 53. Maybe 8%? Okay. Possible. 37. 13. That's great. That's great. Oh, okay. Okay. I see a question on what is a reduced genomic strain? Or 53% is your answer. Okay, fine. The reduced genomic strain is, okay, let's say you have a cell which has like in their genome, they have a lot of things. Like they have an operon system. Also, they have, let's forget the operon system. They have, let's say human cell. That's a very good example. I'm sorry. Let's say human cell. They have, they produce a lot of stuff. They produce antibody, they produce hormones, they produce the usual proteins, they produce, and when they produce the antibody, they do some sort of recombination where they produce each of the segments of the antibody first, and then they shuffle between them. To do all of these things, the human genome has to be large enough to house all of that. But the bacterial genome, they don't care about that. They don't do a lot of the things that the human genomes do. So when you look at the bacterial genome, you find that the bacterial genome is way, way, way smaller than human genome. And the fun fact, since it is smaller, you have to work on it a much less than you have to do on human genome. You want to change one part of the human genome, you have to take care of 26 other parts. But in case of bacteria, maybe that will be just 2 other parts. So that's what I mean by reduced genomic strain. Okay, we have a lot of guesses. So let's just go on with the answer. Whoever said 37%, they can pat themselves on their back. Okay, so let's move on to the next one. So we talked about the design, build, test, learn cycle. And now we are going to talk about things in detail. So the design is where your objective is to generate genetic diversities. So most of the work in the design skill is where you work with the computer. Yes, Abhijit.
I have a question. Can I ask
Yeah.
now? Or... Okay,
Yeah,
so
you can ask
my
right now.
question. So my question is, how would they engineer, like you said the 37% is the right answer, right? How do they do this? Do they, every time they delete one gene, try to grow it? If it is growing or not, then that'd be a lot of work, right?
That's a good question. How did they come to know about the 37%? Okay, much of it is a computational prediction, okay? So you can understand that there will be some genes, of course, which will be lethal to delete. Like you cannot just take a whole chunk of their immune system out. They have the CRISPR system. You cannot just take that out. That would be, they would be viable for the viruses attack and something like that. There will be some metabolic processes which you cannot stop. Like say, for example, glucose metabolism. You cannot stop that. So these are some things that are like lethal for the cell if you delete the gene. And so putting all of those genes that you cannot do anything, that sort of makes up for the rest of the 73%. So the 37% is something that you can anytime delete and the cells won't mind. So that's something more of a computational prediction. And of course, all these years of genetic engineering, genetics, doing genetic science on E. coli has sort of empowered us with the biochemical knowledge of what is actually happening there. So
Okay, thank you.
Thank you. Thank you for asking a good question. Yes, say.
Okay, thank you, sir. When you explained, you said the bacterial cell does not really care about the large amounts. I don't know if you understand what I'm trying to say. Then you said the 37% is what we need. I'm thinking since the bacteria does not really need the 30, large amounts, should we not go for the smallest percentage? Like, since it's going to work. I don't know if you understand my question.
Of course. I mean, like, you should always go for the minimal change inside the cell. It's not like you can manipulate 37%. That means you will completely do the 37%. No, no way. You should always keep it in mind. The minimum amount of work you can do inside the cell to get your result is the best. So that's where choosing your chassis comes in. You may not want the E. coli even if you want to do things in bacteria. The reason being, you want to do the minimal work always. So it is very important to focus on your design part to get the most of it out so that you don't have to suffer an ill fate. Well, to research, sometimes you'll have ill fate, but you don't want to suffer that while you are doing on the lab bench. Does that answer your question?
Okay. Yeah, thank you.
Yeah, thank you. So let's come back to the design part. So the objective is to generate genetic diversity, and there can be multiple strategies. Some strategies can be rational, like you want to do some very defined and specific edits, like that particular point in the genome. I want my edit to be there. So that's rational. Well, what is semi-rational? It's hypothesis-driven, and it may include a lot of enzyme variants. Like you want to change the function of an enzyme, like you want to make it more powerful, but you don't know exactly where you want to do your mutation. But what you know is what is the active site of the enzyme. So what you do is multiple mutations at the active site, one by one. So that is semi-rational. You know your target, you know your hypothesis, but you don't know exactly what to change. So you were sort of focusing on a very short patch and trying to generate the best result. And what is random? Random is you really don't know what you are aiming for. You're just randomly mutating the cell, doing all sorts of harsh mutation and trying to see whether the cells survive that or not. Does anyone really do that? Of course you do. If you want to find out novel things like novel antibiotics, or say, does the bacteria have a very novel DNA repair mechanism? I'm going to do a random mutation and see whether the bacteria survives. It's my hypothesis that this particular bacteria X might have that repair pathway, but I'm not sure yet. Let's just do first round of screening. That's where you do the random. And of course, the combinatorial approach is often more helpful. So if you mix a little bit of everything, that's going to give you the best result. Design throughput. So design throughput is where we understand that all the kind of design and the kind of throughput that we get outside of it. So that can be computational, like a Rosetta protein design. That can be active site mutagenesis, where you plan that what if I change this part of the protein and I change it in computer, put it in some program and see what is the structural change. There can be computational strain design. There are multiple tools for design strain computationally, and then there are tools for pathway prediction. And the very end comes the cis-regulatory modulation. Cis-regulatory means modulation that is happening not exactly where you are changing it, but at the other side. So promoter, if you change the promoter will affect the gene. So that's the kind of cis-regulatory thing we are talking about. So we talked about top-down and bottom-up approach. And what are those things? So top-down is where you have your starting material as a cell, a chassis. You know that I want to work with bacteria. I want to work with E. coli. That's my chosen thing. So what do I do next? I make changes in their genomes, and I do this, I do that, I want this, and ultimately I get this. So basically, you're starting from here. You have a cell, you're starting from there, and you're going down from the cell to the genome to the protein. You're going down one by one by one. So basically, the features are to reducing the genome by removing the dispensable genes. Knockout strategies may be helpful. And of course, if you want to do things like that way, the plasmid and the linear DNA-mediated processes are going to be your most trusted friends. And remember always, it is limited by your starting organism. So if you are starting with whatever cell you are starting with, that's going to be with you forever. What is bottom-up approach? It is that thing where you start with, not with the cell, but with the DNA material itself. So what you do is you start with your computer. You design what you want to have. You design the DNA parts one by one by one. And then you assemble the DNA parts into a complete genome. And then what do? You select a cell. You take their genome completely out of the cells, make the cell void, and now put your synthetic genome inside that cell. So basically, instead of starting from here with the cell, you started from here with the genome, and then you gradually moved up. So that is bottom-up approach. The de novo DNA synthesis and assembly is the key major part of the thing, and you completely reconstruct the whole genome for that. As for the very last part of the design thing, I would say always remember that you need to convert your design phenotypes. What does it mean? So you are designing things to get something, right? To achieve something, an effect. What is the effect? Maybe you want a high level of transcription. Maybe you want a high level of translation. Maybe you are looking for you are trying to make a cell-based biosensor. So you need to be really careful about the protein-protein interaction, because the biosensors will catch something. It will relay the signal. So you need to be aware of the protein-protein interaction. But how do you want to measure it? You cannot just go inside the cell and measure all of these things. So you need to convert desired phenotypes into measurable parameters. That can be growth rate. That can be survival under selection, like antibiotics. That can be some sort of fluorescence. Your protein interacts with another protein. And maybe some of the protein, you have tagged them with GFP. So whenever they interact, there is a GFP signal. So these are the kind of measurable parameters you should always put inside, so that the desired phenotype can be really and easily measured. Before we end the design side, I would say there are a few computational designing resources, and not that you need to be super aware of it, but if you in your free time you want to play with it, most of them are almost free. Like you probably already know about the UniProt and the NCBI NLM sites. Those are great resources for proteins and nucleic acids things, and you can basically get everything, know everything about a gene, about a protein from these resources. There is this LCSB database, which is a very, very interesting database for strain engineering. Let's say, for example, this Shiki factory inside this LCSB database, it tells you about all the possible modulation from the shikimic acid pathway. And shikimic acid pathway, let me tell you, is a very, very important pathway for designing drugs. So, and also you have vector builder. At any point of time, you want to optimize your codons for E. coli or for other organisms. Vector builder is a very good resource. Even if not for codon optimization, you want to play with like how to find restriction enzyme cut sites within the genome. Put the genome, take as for your home exercise, you can do something. You can take a gene or a protein from the UniProt or the NCBI and put that into the vector builder to see whether I'm getting, whether you're getting like, whether your nucleotide sequence has any sort of restriction enzyme cut site within them, or maybe you want to design a primer, and you can go ahead. Of course, the first attempt won't be 100% correct, but no first attempt is. Go on, play with this. So with that, we move on to the build cycle. And for build, what you need is the tools, right? You want to build a genetic mutation. You want to build a genetic modification. You need tools. Now, what are the tools? The tools are mostly of two types, nucleases and mutagens. So nucleases are the things that will cut your nucleic acids. Mutagens are the things that will change your nucleic acid. So these are the very important tools. And there are multiple variations of these things. The goal being editing genome in a programmable manner. And do not ever forget this, that your edit has to be programmable. Unless and until you can program this, like you make some changes in the genome, but you wanna be able to put a leash on it, like at any point of time, by any small modification in the growth medium or any small modification in some outside inducer, you can program that change. Like the change will be there, the change will not be taken away, but you can modulate whether to produce the protein more or to produce the protein less. And editing can be precise or holistic, like it can be very specific, or it can be like all over the genome. But what is important is to know about the cost by throughput ratio. It should always be lower. What does it mean? That the cost of your edit, like you wanna do a whole lot of edit, that's fine. But maybe your project plan is to do 10 different kinds of genetic edits to get one protein. No, my friend. That's not really a good idea, because at that point of time you are spending so much money on doing those 10 kinds of genetic edits. What are you getting? Just one? No. So you need to design your things in a way so that it's cost effective. Your cost should always should be lower than your throughput. So talking about throughput, the build throughput is in a different ranges. Like if you are going for multiplex gene assembly, like you have different genes and you make edit in one gene, then you make edit in another gene, and you then assemble all of them together. If you're doing things like that, you can reach the throughput up to 10 to the order of 10 to the power 6. You are doing degenerate gene synthesis, where you don't care about whether the genes are in array or not. It is degenerate, and then you are assembling it. It can go up to another order. Multiplex oligosynthesis, it goes down in the order. If you're doing error-prone PCR, where error-prone PCR is a very good tool for introducing mutations inside your specific segment. So you are doing the PCR, but instead of being a very high efficient polymerase, you are using a polymerase that would introduce errors. Or if you're doing multiplex genomic engineering, you can reach up to 10 to the power 10 order of magnitude. So these are important things for knowing your throughput, something that you should care about if your design is to end up in industrial scale. So in order to know the tools, let's talk about some useful tools for build purpose. So the first thing that we need to know is gene cloning. Maybe we know a lot about this, but then it would be a quick recap. But if you'd like to ask specific questions about this, feel free to ask in the chat room or raise your hand. So here we have a picture of a very small part of the E. coli genome. And we can see that the strip A is the one that we want to clone. We can use the nucleus to chop the genome into multiple parts. But how can we select the one strip A segment? That is where the polymerase chain reaction comes in the picture. So what is polymerase chain reaction, or PCR? We are probably all aware of this, that where you have a DNA template that you want to replicate. And what you do is you need a whole lot of nucleotides, primers, and the polymerase itself. First, you denature the strand so that the strand is single-stranded. Now you attach the primer to it. We call it primer annealing. And if you are aiming for a cloning purpose, you can have specific restriction enzymes attached with the primer. You probably know that already. So this is how the polymerase goes on, make another copy, and so on and so forth. And ultimately, you get your PCR product, which has the primers annealed at the end. And these primers may be very important for your cloning purpose. So the cloning strategy, and the cloning strategy is as it goes. Here you have an example of a cloning assembly, not just a cloning strategy. So if you look closely, you will find that the first plasmid, which has a GFP, has has 4 different restriction enzyme cut site: E, X, S, and P. And these E, X, S, and P are present in all the other 2 plasmids as well. What do you want to do? What your goal here is to take GFP from this plasmid, the origin of replication from this plasmid, and the chloramphenicol gene from this plasmid to produce something like this. So these 3 different things, these are like bricks that would rise a home, that would build a home. These are called biobricks. And the process by which it is done is called a biobrick assembly. So it is a very specific kind of cloning strategy. What you're doing here is you are cutting this first one with E and S. You're cutting this one with X and P. And you're cutting this one with E and P. Okay, fun fact. What is common between them? P and P. So of course, their P end and their P end, they can be ligated together. What is common between these 2? E and E. So their E end can be ligated together, and that has happened. But if that is so, what about their S and what about their X? How is that getting along? So S and X, for that particular position, you choose a restriction in 2 different restriction enzymes, but the beauty lies in the fact that both of the restriction enzyme will produce a complementary overhang. So the overhangs are reverse complement to each other, so they will just anneal as it is, but they will not regenerate S or X. They will regenerate something completely different. So by utilizing very specific set of restriction enzymes, you can do all these sorts of things. And these are, if I'm not wrong, biobrick assembly sort of came into the picture 15 years ago. And from that time, it is still one of the most popular methods. So if we want to use this sort of biobrick sort of thing, what you need to do is take control of the computational resources that you have. And one of the very great resources is the benchling. So maybe some of you are familiar with the benchling. Some of you are not. Here I have, I'm showing you the PBR 322. It's a very common adgene plasmid. I'm showing you the map in the benchling. You can take any plasmid, you can download the plasmid from adgene, or you can download them from anywhere else and put them into benchling to get the complete map out. And then you can see that there are the restriction enzyme cut sites that are mapped alongside the sequence. So you can have a pretty good idea if you want to use the HINT3 or the EcoR5, where exactly they're going to cut. It's very simple to look, just have a look at it and you will know everything. So benchling is a great resource. Also, for a quick recap, I would say look at these things where you have like these are very important points for the benchling file, where you have different things. So one thing shows you the molecular features of the nucleic acid segment you have added. Another one, it helps you to select enzyme for your restriction enzyme digestion. It helps with primer designing, sequence alignment, and also if you want to design your CRISPR based on your sequence, you can do that in benchling. As a matter of fact, I'm just showing you an example of the restriction enzyme tab. So once in the benchling, if you keep that click that tab, you will see that there are a whole lot of enzymes that are getting listed here. And if you select the Eco R5, it shows you what is the cut site and what is the activity, what kind of buffer you want to use, all these sorts of stuff. So very useful stuff, I would say. Yeah, somebody in the chat also said, Murimi, that benchling is also a pretty good resource. And thank you. I did not see it beforehand, but I hope I have acknowledged that. So there is also a link posted here, and once after this lecture you get the slides, you will know you can access the link, and this is a very good like reading material for how to digest and ligate using the benchling. It's a very easy step-by-step process. Go ahead with it, and once you have and the fun fact of computational resources like this, that you can play with them in your leisure time, and you can be a master of that once you go to the lab. And believe me, if you have mastering control over the computational resources, your lab life is going to be so smooth. Okay, let's move on to the next thing. Now you have cloned DNAs, but do you really, I mean, once you have cloned the DNAs, do you really want to just clone it and go? Maybe not. Maybe you want your DNA parts to like joined together. So DNA assembly method refers to a procedure of physically linking the multiple fragments of DNA. And there are three main types. One we have already discussed, the BioBrick standard assembly, which utilizes the restriction ligation system. The other one is the type 2S restriction methods, such as Golden Gate or Moklo. And the third type is the end homology methods, such as Gibson assembly or infusion. So I would not go like in really good details of it, but just to say the first one, restriction ligation, the restriction enzymes are all type 2 restriction enzymes. Type 2S restriction enzymes are different from type 2 in a way that type 2 has a very specific cut site. They will always cut at a very specific position within their recognition sequence. Type 2S does not do that. They have a recognition sequence for sure, but they do not cut at a specific site. They always cut 3 or 4 nucleotides away from the recognition sequence. So in a way, they produce a very unique overhang because you cannot control those 3 or 4 nucleotides. That can be anything. That can be any damn thing. So if you are using one particular restriction enzyme, you can achieve multiple effects with that. Let's talk about Golden Gate as the system which utilizes type 2S restriction system. The BSA1 is a type 2S restriction system. And you can see that BSA1 cuts right outside of their recognition sequence one nucleotide here, and right outside of their palindrome, their reverse complement, 4 nucleotides here. So the cut is like this. So at any point of time, you will have a 4 nucleotide overhang in the 3' to 5' strand. So if you are using BSA1, you can basically do a one-part reaction. You can put everything within a single part. The restriction enzyme, the ligase, the ATP, the buffer, everything, and put it in a cycle where the first few hours, you maintain the temperature that is good for BSA1 digestion. And then for a few hours, you maintain the temperature that is good for ligation. So, and once you do that, you get the product in just one go. You don't have to go for like BioBrick thing that I want to cut this and ligate this. I want to cut this and ligate this, and then I want to ligate this. No, you can do this in like one part. And you know how many people have seen that you can actually do 50 different fragments of DNA and assemble them using this kind of thing. So that's great. So if you ask me, why is Golden Gate so popular? Because it's one-part assembly. You just need one restriction enzyme and ligase. It's rapid, it's high efficiency. It assembles many parts at the same time, and it's scarless. What does it mean, scarless? Let's go back. So since you don't have very specific nucleotides at these positions, the ends, once the ends are joined, they produce a site that would be totally unrecognizable as something has got it beforehand. So you will not have any scar left from your nuclease treatment, your restriction enzyme treatment. So that scarless thing is a very sought after technique for many of the synthetic biology purposes. So is there any disadvantage of Golden Gate? Well, there are. The restriction enzyme sites cannot be at internal location. Of course, that's true for any restriction enzyme, but in case of Golden Gate, you don't want to have another BSA1 site within your sequence. And it's sometimes difficult to find compatible 4 base pair overhangs. You can identify, if you can understand, if you want two things to join, to be joined at the ligation state, you want their 4 base pair overhangs to be exactly reverse complement to each other. And you need to find out that sequence. You need to get that sequence inside that. It sometimes becomes very difficult. So in order to do that, what you need to do is PCR with a primer that has that 4 base pair overhang. In that way, you can attach the 4 base pair overhang and the BSA1 cut site to the borders of your gene of interest, and then cut it, then get the perfect 4 base pair overhang that you are looking for. Why? Because maybe your vector has those 4 base pair overhangs. Modular cloning or MOCLO is something that is based upon this Golden Gate principle. But where it differs from the Golden Gate principle is that it has different levels. At the level 0, you have, here is an example where you have a different DNA assembly of promoters, 5' UTR, signal peptides, coding sequences, and terminators. Remember, these all are different DNA assemblies. T1, one terminator, T2, another terminator, like this. What you can do with Golden Gate is that you can take P2, U2, SP2 series to T2, make a complete unit. Like that, you can take P1, U1, SP1, series 1, T1, make a complete unit. And all of these units can be joined together among themselves at a multi-gene, as a multi-gene construct. And now what you can do is you can put the BSA1, using the BSA1 site at different ends, you can clone them all together within one particular strain. So that's basically what you're doing, you are assembling different modules by utilizing the Golden Gate principle. So that is what modular cloning is. It is nothing different. It's just knowing Golden Gate principle and putting that into a different design. That's it. So now that you have, now that you have the cloning part done, what do you need to do? You need to put your clone inside the cell. And there are two ways you can do that. You can put that as a plasmid, extracellular material, extragenomic material. I'm sorry, not extracellular, extragenomic material. Or you can do that by integrating it within the genome. And if you want to integrate it within the genome, you need nucleases, spatial nucleases that would do that. And there are two types of nucleases that does that. One is a protein-based recognition. One is a nucleic acid-based recognition. What do I mean by recognition? How are they recognizing their target? Recombinase, integrase, zinc finger nuclease, there are other, talents, and many more. What do? They are a protein molecule, enzyme molecule, which goes on and recognize a particular genomic region that they want to integrate inside your target. For the nucleic acid-based recognition, you have recombinering, MAGE, CRISPR. All of them, they do what? They go on recognize a particular region in your target, but with the help of a nucleic acid. Let's say for CRISPR, you cannot make a CRISPR selective without its guide RNA. So that is the nucleic acid-based recognition. On the other hand, the recombinase integrase, they have a protein-based recognition. So a quick look at the integrase recombinase function. So these are very directional, and you don't have to go to the like very intricate details of it. But if you look at here, you will see that there are some attachment sites. Hello? S23 Ultra. Are you saying something? If not, I would request you to mute your mic. Otherwise, if you're saying something, it's fine. You can just be a little bit louder. Okay. So with the add P sites and add B sites, what you can do is that you can put the integrase, and as I said, that these things, they are protein-based recognition, so they need to know about these sites. You need to have a specific site at which they can integrate your thing. So let's say this add B site is there in your genetic modification that you have made, and this add P site is there in the target genome. So then you can sort of make that integration happen with the integrase. If you want to do genome engineering with that, you can do it at a multiple level. You can have a donor vector which has the @B site, where you have your gene of interest and your marker, and the docking site in the genome has an @P site. So once you apply integrase, and there are two different type of examples with the two different type of integrase, where what you can do is once you apply the integrase, the marker and the gene of interest are there in the genome. So you have a complete genomic integration. The fun fact is that depending upon what kind of integrase you use, you can have different directionality. So you can have the marker and gene of interest in this example just as it is, or you can have the promoter 1, gene of interest, promoter 2, just as it is, depending on what kind of integrase you're using. It can flip the direction. So the @P site is something that is there because of the fudge attachment. It's a very evolutionary thing present in bacteria, and @B site is something you are putting in your donor vector. CRISPR. As for the CRISPR, you have this is probably the most advanced technique that we have right now, and there are multiple researches going on this one. For CRISPR, what you need is the enzyme identifies portions in the DNA by the guide RNA. So the guide RNA pairs with the DNA at a specific site, and the CRISPR recognizes that site, and the CRISPR has a nuclease activity, so it cleaves at the RNA-DNA junction, and then it repairs the whole thing by the DNA break repair pathway. So there can be different pathways that can be employed for the repair thing. It can be NH-EJ pathway, or it can be some other pathway depending on what kind of CRISPR system you're using. So we know that CRISPR is there, and its functionality depends on the guide RNA. If the guide RNA, so you can change the guide RNA, right? You can say the guide RNA go and bind to DNA region 1 in the genome, and the CRISPR will go there, and it will recognize that position and cut it. You can say guide RNA, go and bind to the DNA region 2. And how can you introduce mutations with it? Well, of course, if you introduce the mutation in the guide RNA, the CRISPR will try to incorporate that as well. Quick challenge. What is the best way to improve CRISPR-Cas editing system? Make a guess. You can make your CRISPR less selective. You can fuse your Cas9 to a reverse transcriptase. You can remove the RNA-based selection. Let's make it DNA-based selection. You can fuse the CRISPR with a recombinase or an integrase, for example. What do you think is the best way to improve the CRISPR-Cas editing system? I'll take some time so that you can mention your results in the chat. D, okay. People are thinking that it's fusing with recombinase. Oh, okay. A few minutes, and then I'll give you the answer. B, maybe C. Okay. Answer is B. Why? Because you have the guide RNA. If you fuse the Cas9 with a reverse transcriptase, what a reverse transcriptase does is that it makes DNA from RNA, right? So basically what you have is an RNA molecule, guide RNA, and it can make the DNA. You can incorporate your edits, your mutations inside the guide RNA, and the reverse transcriptase will do exactly that. So you don't need the DNA break repair pathway to take care of your repair thing. You can use the reverse transcriptase with that, and you don't need to rely on their machinery to incorporate your mutations. And feel free to ask questions about this in the Slack channel, and I'll be happy to answer you guys. Oh, yes, Abhijit.
Yeah, so like, what is the difference between B and C?
Okay, the B and C is where, in case of B, you are fusing the Cas9 with another protein component, the reverse transcriptase. And in case of C, you're removing the RNA-based selection. You are making specific edits in Cas9 so that it does not depend on RNA anymore. Maybe it does not require a nucleic acid part anymore. Something like that.
Okay, understood.
So this kind of editing that I just told you, fusing Cas9 to reverse transcriptase, it is real. It is called prime editing. You can go ahead and look for it. It's been a really great paper for quite a long time in the last few years. We can do directed evolution. So like, are we good at engineering biology? If we ask this question, the answer is arguably no. So what if we let the natural mutation do it for us? And what do we mean by natural mutation? Natural mutation is where mutation is creating variations, and unfavorable mutations are selected against, and the favorable mutation stays along. So what if we do that thing inside the lab, inside our chassis? That is called directed evolution. You are letting the natural mutation do its own thing, but you are directing it to a specific way. And how are you doing that? That puts us with another technique called multiplex automated genome engineering, or MAGE. What you do is very simple. You have an input population, you introduce some single strand oligos. Yes, proceed.
Hi, taking you back to CRISPR-Cas. one of the options was making it less selective. I wanted to inquire, is CRISPR-Cas selective, or can we make it more selective? And if we can, what do you think are the ways we can do that?
Well, you can make CRISPR-Cas more selective. There are ways, actually, there is research happening on that. It's a very good question. CRISPR-Cas, you can make CRISPR-Cas more selective by just putting, I mean, not by just, but you can do that by making so CRISPR-Cas, the Cas9 protein itself has two domains. One is the nucleotide cleaving domain. Another one is the nucleotide binding domain. So if you can engineer the nucleotide binding domain to make it more specific, like maybe, and just as a matter of example, it's not exactly as it is, but maybe the CRISPR, the Cas9 nucleotide binding domain recognizes at best 4 nucleotides and binds them. It sees like a match with the RNA and the DNA, and it binds there at 4 nucleotides. Maybe if you increase it to like 8 nucleotides, it will be more specific. It cannot go any wrong way at all. You are providing it with a lot of room to look for correctness. This is one way you can go on with that. I hope it answers your question. What was talking about the multiplex automated genome engineering, MAGE. MAGE is a very important tool for doing genome engineering at scale. All of the genome engineering thing we have talked about till now, they are like lab bench thing. You can do this thing, and then you can make a bigger thing. Then you can make a more bigger thing. But in MAGE, what you are doing is not you are not targeting one bacteria or like a tube full of bacteria. You are targeting a whole like tank full of bacteria. So your input population, you are providing it with single-strand oligos, the oligos that have mutations. The oligos will be taken up by the bacteria. Then those oligos will go inside. Those oligos will be attached with the genome. And since they have mutations, some of the bacteria won't survive that mutation. Some of the bacteria will survive that mutation. And once you are done with the whole round of growth, you can select the bacteria that has survived, and you can go on with that. So you are basically directing bacterial evolution with this cycle. You can do a whole lot of things, like you can do transcriptional engineering or translational engineering. Like you can play with scale, like not just one protein, you can go for like 10,000 proteins at once with this kind of thing. And with that, we come to our test cycle. And the test cycle is all about testing your throughput, testing your productivity. And there is 2 things that you need to remind yourself of. That one is scale, that we have talked about, that you can do things at plate reader, but you need to go at this 200,000 liter scale. And it has often been seen that a plate reader to 2 liter is the most critical step. The success rate is only 15 to 30%. But once you have achieved this 2 liter thing, you can go to the 200,000 liter thing in a success rate of 70 to 90%. If you ask me, why does that happen? It is because when we do things in lab, we often don't consider, or often cannot consider, things that are outside of cell. How the cells are being affected at an environment in a bioreactor. We don't get to know that. So we always need to get our design from the lab bench to the bioreactor if we want an industrially important thing to produce. The second thing that you need to remind yourself of is the kind of technique you're using for your test. You can use the droplet-based microfluidics. You can use UV visible spectrophotometry, fluorescence, or you can use chromatography. But what are you trying to achieve? You're measuring quality. What kind of quality you are trying to achieve? If you want high quality but low number of scale, you can go for chromatography. If you want a high number of scale and high quality, you can go for mass spectrometry. So things like that. So test throughput is what I just talked about, that if you are choosing for different kinds of techniques, you need to know which technique would give you what kind of scale. So there is a bottleneck in testing using traditional approaches. So let's have a look at the classical traditional testing. This is a workflow of a classical traditional testing where you have a mutant library, you have made some designs, you have put that in a tube, you have grown the cells, you have gone for the production. And now you're testing, your production is doing okay or not. And the order is in 10 to the power 3 to 10 to the power 4. The next high throughput testing is where you employ the robotics. You employ a liquid handling system, you go for the production next. 10 to the power 4 to 10 to the power 5, you have one more order of magnitude. The next is the ultra high throughput testing, where you are employing the fluid mechanics or the fluorescence activated droplet mechanics. So fluorescence gives you a whole lot of power to screen a lot of things at once. The other thing that gives you that kind of power is sequencing. So here are some measuring instruments that you can go through that you will commonly find in your lab, and there is a comparison of their throughput and stuff. You can look at them at when you look through the study material. It's not something that you need to keep it in mind, but of course, if you are trying to achieve something in a scale, you need to choose your technique that way. As for sequencing, there are two very popular approach that is happening at this point of time. One is RNA sequencing, another is barcode mapping. So RNA sequencing is basically where you make cDNA from your RNA. So once you have your design done, once you have your build done, what you are doing is you are taking the cell that is, you are saying that it is producing my thing. I want to achieve protein X. I want to know whether, or maybe I want to achieve protein X, Y, and Z. I want to see whether the cell is producing that. What do? You take all the RNAs the cell are making. You take all the mRNAs. So you will have the mRNAs of protein X, Y, and Z, right? So you have designed your protein sequence in a way that the RNA part of it will have a barcode attached to it. Now you make cDNA out of that RNA, and you sequence the fragment ends that has a barcode. So now you can read those maps, and you can say whether the protein is getting produced without moving to the protein part. What does it do? It gives you a very fast and very accurate result of your test. And two, it allows you to know whether you are having any problem at the transcription level or not before you move to the translation level. As for the barcode mapping, you do the same kind of thing, but not with the RNA. You have a mutation at some point, and you want to track it. You make a barcode. You make a barcode at the very end of your DNA. And you make different barcodes for different things. For mutation, you use one barcode. For no mutation, you use another barcode, something like that. And you then amplify the products. And once you have your products, once it is inside the cell, you take all the genome of the cell out, do a PCR, and go for sequencing. And once you have, you know which barcode attached with the mutation. So once you subtract that barcode, you get everything rest. So that is a very specialized type of sequencing where you are not sequencing the whole DNA. You are just sequencing the barcode. So you have to sequence a very short patch, let's say 30 nucleotides or 24 nucleotides, something like that. And you will know everything about the gene because you haven't changed anything else in the gene, right? You just introduced one mutation. So in order to know that mutation, that barcode is enough. You can do cellular screening, like antibiotic-based screening, where you couple the function to the growth. So in case of antibiotic screening, what you're doing is that maybe you know that your cell produces a highly functional antidote that blocks an antibiotic. Maybe your cell is chloramphenicol resistance, but how do you know that? You grow them in presence of chloramphenicol. And if it survives, you know that your design has worked. So here you are coupling the function to the growth. You can couple the function to the fluorescence, where you want to see the production of a protein that has been tagged with GFP. So every time the protein is getting produced, the GFP is fluorescing. Or maybe you want to track an enzyme which is activated only when the repression from its promoter region or operator region is off. And the repression is only off when the fluorescence is there. You can screen everything. So you don't need to go for sequencing, getting the gene out of the cell. You can have the cell intact, and you can see your productivity testing by measuring the growth rate or by measuring the fluorescence. And these are the examples where you have converted your desired phenotype to a measurable parameter. And that puts us to learn cycle. And what is there in the learn cycle? The learn cycle is about lessons. The lessons being fitness is paramount for manufacturing success. So you can do a whole lot of engineering, but the engineering has to be sustainable within the cell itself. The cell needs to be happy with that engineering thing. Cell needs to be fit. Fitness is very important. Always remember, it is harder to engineer which is something that is already optimized. If your strain is like highly optimized, then if you change one thing, the whole thing will just fall apart. Remember the 37%. Since you have the advantage of 37%, you can do everything. If the 73% was everything, 73% was the 100%, you couldn't have done everything. Like you change one thing, the whole cell will die. Choose a chassis that is not highly optimized in terms of genetics. Epistasis or epistatic effect. You change something here, some other change is getting somewhere else. This can improve the stacking mutational effects, but it's not rational. Like, let's say in your design, you have introduced a mutation unwillingly, but then you change somewhere something that would completely negate the effect of this mutation. It's possible, but it's not always rational. So be very careful about this kind of epistatic effect. The lesson for being, of course, highly diverse designs are key to higher fitness. The more diverse your design is, the more fitness you are going to get, because the more evolutionary advantage you're providing to your chassis. But always remember the final thing: use the takeaways from learn to design better. We are at the very end of our lecture, and I'll just show you one example of a learned thing: bacteria to cure cancer. I'm going to say this, that my slide has one more example, but I'm not going to cover that. That example is just an example. You don't have to remember that or go through it in very detail. But that example is about bioremediation, how oil pollution can be was cured a long time back using Pseudomonas putida. So I want you to read that by yourself to have an idea how we can tackle real life problem using synthetic biology and strain designing. How we did make changes in the Pseudomonas putida. There was a guy working in the General Electric company named Ananda Mohan Chakraborty who first did this thing. And it's a very interesting thing. You can find it in internet. And oil pollution is still a very important thing in our world. And so maybe for your project or in your future, you want to have some idea from that. So go ahead, read that. For now, for our end, we have this: the bacteria that cures cancer. We know about Salmonella typhi. It's a very pathogenic bacteria. It's a danger to human. Why? Because it goes inside the cell. It can reside inside the cell, and your body immune system may not be able to find it. Researchers with synthetic biology has made best use of that. What they did is they took the pathogenic genes out of Salmonella, and those falls in those percentage that you can modulate. So it does not affect Salmonella at all. So it does not matter whether you are taking this out or not. Salmonella is happy with that. Then what they did, they made a whole lot of genetic engineering to produce acetyl salicylic acid, which is the precursor of aspirin. And what it does, it converts the salicylate. It gets converted to salicylate. And salicylate or salicylic acid then converts or activates cytosine deaminase. Cytosine deaminase makes 5-fluorouracil. 5-fluorouracil is a highly potent toxin for any kind of cell. It messes up with the DNA replication. It does havoc inside the cell. So if your salmonella is targeted for the tumor cell, you make your salmonella in a way that it would only recognize tumor cells and goes inside them. Tumor cells are growing cells, right? They are always growing. They want to have more food. They would get salmonella inside them. Salmonella will go inside them. And then 5-fluorouracil will kill the tumor cells. That is a research that has already been published. You can go to the link, you can read about them. So, and that, I hope, should encourage you to think about all the beautiful things we can do with synthetic biology, we can do with strain engineering. With that, the lecture ends. Thank you.
Thank you too.
Thank you.
I'll stay a little longer. If you have any question, you can ask me here. You can ask me in Slack later. Whatever, you can ask me any question about the homework in the homework channel in the Slack. So. Oh, Prosi, have you raised hand again? Or is it from the previous? Yes. Go ahead.
Yeah, just an inquiry. We were talking about how much of an organism's genome we can manipulate. How do we determine that? Are there any tools we can use to determine that? Yeah.
Very good. So the best possible tool is probably the computational tool. I would answer your question in two ways. One is if you are starting your work with a known organism. Two, if you're starting your work with an unknown organism, a novel organism you have chanced upon. So if you're working with a known organism, you should always go for literatures first to know what people have done, because people have been doing this for a long time. You would have a very good idea what kind of thing I can do in there, what kind of thing I can play with them, and what I cannot play with. Second is to go for the computational tool. There are some computational tools that I have listed at the design section. If you go there, you would have resources that would tell you the complete genome sequence or the complete genome features of different organisms, known organisms. So from there, you can learn. It also depends on a little bit of common knowledge. Like if you look at a genome feature, you can see that, okay, fine. These bacteria or these yeast, they produce salicylic acid. What if I provide salicylic acid in the growth medium? Then I can go ahead and take the salicylic acid pathway from there. It does not need to provide salicylic acid if it is producing salicylic acid as a metabolite. If it is producing salicylic acid as an intermediate of something else, then you cannot delete it. Then that else is going to be affected. So you need to know that. You need to know the whole metabolic pathway. That does this pathway 1 connects with pathway number 8? If so, can I make changes in pathway 1? in a way that it would make change in pathway 8 or not make change in pathway 8. So these kind of things you need to consider. But what if it is producing salicylic acid just for its own nutrition? Delete that pathway. Put salicylic acid in the medium and you'll be fine. So these are the kind of thing you can do. For unknown organism, of course, you need to go for a lot of trouble. Like you cannot start doing synthetic biology on an unknown organism on a very first attempt. You need at least some years of knowing its genome, knowing its metabolism and everything to make it a standard organism. That's why actually synthetic biology people, they don't want to do anything with novel organisms. They want to create novel organisms out of the standard ones.
All right, thank you. That was really nice.
Thank you.
Thank you.
Okay. If anyone else does have a question, I'm gonna be here for a couple of minutes, 1 or 2 minutes. Otherwise, you can always ask. Well, of course, whoever he's here till now, I would say the homework will be released. Go and have a look at the homework. The homework is a bit of what you have understood from this lecture based on that, and also based on what are the kind of exercises you're supposed to do. So please play with the vector builder, play with the UniProt. I'm not sure whether you will have access to Benchling, because Benchling usually requires an account, but if you can do things with your free account, go ahead, do that. That's a very, very utilized thing tool. So go ahead, play with that. 1 or 2 of the homework questions is based on what you can do with UniProt, what you can do with vector builder. So I would encourage you to do that. Because and any kind of problem you have, like if you even have problem with like navigating through the online tool, please post it in the homework channel in the Slack, and I'll take some time out to guide you through that. Well, Dan, you are totally right. Yes, Billy, I'm coming to you just after a couple of minutes. So Dan, Dan said in the chat that I think deleting less essential genes may bring about changes in the organism's phenotype that are not immediately obvious. And you are right. It's not going to happen every time, but sometimes it is true. Sometimes you make changes. Okay, I'll share a story about this. I used to work with Archaea in my PhD, and I was doing enzyme biotechnology. So I was making changes inside the enzyme, but I was not doing the work with the enzyme within the Archaea itself. I was not doing archaeal cell biology. I was cloning the protein from there and putting it in E. coli. So all of these things, I was making small edits, small mutations inside the enzyme, so that because it is an enzyme that binds the RNA and then exerts its function. So I was making changes in the RNA protein interaction domain. And I found out that while I was doing that, I made a mutation without realizing. So it's a 2 protein, 1 RNA complex, and I was working with 1 protein of that complex. And without realizing, I have made some changes in its RNA binding domain that somehow the protein is not binding to the second protein at all. And that is, I mean, that became a point of investigation in my PhD. But that is something you don't understand at first, because everything is happening fine. It's just that it's not binding to the second protein. So maybe there are some things and you have said in phenotypes. Well, true, in phenotypes, it is very difficult until and unless it is a very prominent effect. Yes, Billy, what's your question?
First of all, can you hear me?
Yeah, I can hear you.
Okay. Okay. Thank you so much. I'm quite a novice in this area. I'm a plant pathologist.
Okay.
To start with, and quite a number of things which I think I really need to catch up with, and I'm just hoping that I'll be able to make it through the course, through this particular intermediate one. And I would really need your indulgence. Maybe I'll be asking because I really want to finish. However, I've just gone to Slack, and we don't have that. There is the group making. I'm not seeing the homework. The homework what?
Homework, the homework of the week 3?
There are groups in there. I think they are called channels. I've not seen the
yes.
homework channel there. Maybe.
Okay, let me see. Oh, there is a homework questions. My bad. It's not homework. Homework questions.
Oh, homework questions. I'll try to check it out.
Yeah.
Okay. I just wanted to make that comment.
Yes, sure. And feel free to ask anything at the homework questions. There are 2 channels. One is lecture questions, another is homework questions. If your question is specific for the homework, put it in the homework question. It's for everyone. And if it is for anything about the lecture, you want to know something more about the lecture beyond what the lecture, what I told you, you want to know some more resources, feel free to ask there. As for the final project, whatever Heidi told you, remember that. And one more thing I would like to add: when you are trying to design a project, it does, as Heidi said, it does not have to be like really big. It can be small, but your understanding of the project needs to be paramount. So that's important. And for that, I would suggest it would be really helpful if you search for review papers in Google. So review papers about synthetic biology applications. A review paper always talks about multiple different things. How they did that, how they do that. Find a project, find a work that excites you. Go to their actual paper and then read about how they did that. What are the steps they took? How they made some changes in the genome. Do not copy that project, of course, but try to copy their steps. Like they did design first, then they built the genetic tools second, then they looked for that particular thing third. Go like that. So that will help you to sort of build your own project outline. Your idea is yours. Think about an idea by yourself. But the pathway to go there to achieve something, there are multiple literatures out there in the world that people are doing. Follow them to know if you are doing it correctly or not. Okay, with that, I will say goodbye to all of you. Have a really nice rest of the day, and I wish you all the very best for the rest of the course. Meet you again.